Hello guys, welcome to my channel Education Plus. Today we'll be discussing about protoplast purification and staining. In my previous video I discussed about how protoplast can be isolated, and if you have not seen that video, the link has been provided in the description below. Kindly check it. So let's begin with the lesson now. Now, the enzyme digestion mixture, which was obtained by treating protoplast by enzymatic method, so that particular solution is not pure. It contains subcellular debris, other undigested cells, broken protoplast along with a healthy protoplast. So this purification can be carried out by three methods. We have filtration, centrifugation and washing. So let's go for filtration first. Basically, filtration is a simple method wherein the solution containing protoplast is being filtered using a nylon mesh which is having a pore size of 50 to 100 micrometers. Here, we are removing large portions of undigested tissue and cell clumps. Is done, we go for centrifugation. Now here the filtrate is being treated with sucrose and protoplast suspension medium. Then it is centrifuged at 100x for 7 to 10 minutes. Basically, we take a centrifuge tube containing protoplast suspension medium to which sucrose is added. The sucrose, it helps is, why are we using sucrose, is that it helps in maintaining a gradient, separation gradient, wherein the protoplast will be separated from the other unwanted substances. So this particular filtrate, okay, it is being treated with sucrose in a protoplast suspension medium, and it is being centrifuged at 100x for 7 to 10 minutes. Now after centrifugation, the debris will settle at bottom of tube in the form of pellet, and a band of protoplast will appear in the middle one. Now this band of protoplast, it is sucked off using a Poucher pipette, and then it is transferred into another centrifuge tube. The procedure wherein you can see that there is a pellet. This is a centrifuge tube. To the bottom you can see a pellet of dead protoplast, and in the middle there is a band of protoplast which is being separated, and the solution is a sucrose solution, and the protoplast suspension medium containing sucrose solution, and the protoplast which is separated. Sucrose and protoplast suspension medium and centrifuged again. Now basically this same step we have to repeat it for 3 times. That's why the step is known as washing. What we are doing is we are washing the solution to obtain a pure protoplast. So again, the suspension of protoplast, we are treating it again with sucrose and protoplast suspension medium. It is centrifuged again at 100x for 7 to 10 minutes, and then again the debris will settle to the bottom and a protoplast band will appear at the top. It is again being sucked up with a Poucher pipette, transferred into another centrifuge tube, and the same process is repeated 3 times. Okay, after which protoplast can be purified completely, and it will appear as a band between sucrose and the protoplast suspension medium in a centrifuge tube. Now the protoplast, it is separated from remaining mixture. Now again, how can we go for separation of protoplast? Now from the solution which was used for for the purification of it, the mild traces still remain, so that also has to be removed off. So here the protoplast, it is separated from remaining mixture using 20x percol solution, and this percol solution, it is nothing but it contains 0.25 molarity mannitol and 0.1 molarity calcium chloride in a centrifuge tube. So the tube containing percol solution along with the protoplast solution is centrifuged again, resulting in the separation of mixture, and at the end of it, the protoplast will float to the bottom, and the other unwanted debris will settle to the bottom of tube. Now after obtaining the protoplast, now we have to check whether if it's a viable one or it's a dead one, so that can be assessed by means of staining. Now for the staining of protoplast, 3 dyes can be used. We have fluorescent diacetate staining, it's also called as FDA staining, phenosafranin staining, and chalcofluoride staining. Now first go for fluorescent diacetate staining. Let us see what is FDA staining. FDA is nothing but it's a staining dye. The cells, when treated with FDA, the FDA, it will accumulate in the cell wall of viable protoplast. It will accumulate in plasmalemma of viable protoplast, living protoplast, and these cells, when they are observed under fluorescent microscope, they'll fluorescence yellow-green within 5 minutes. They'll fluorescence fine yellow green. Within the 5 minutes dead ones will not fluorescence. So FDA basically what the FDA is it is dissolved in acetone and it can be used at a concentration of 0.01%. Protoplasts when treated with FDA, they must be examined within 5 to 15 minutes after staining with FDA, because FDA it will disassociate from the membrane after 15 minutes. Okay. The staining method which can be used, we have the phenosafranin staining. The phenosafranin, again, it is a dye which is used for staining of protoplast. The cells, when they are treated with this dye, the dead protoplast will turn red, while viable ones, the living ones, will remain unchanged. So this dye is specific for dead protoplast, while it's not specific for the living one, because the living one remain unchanged. So for this purpose, FDA it is used at a concentration of 0.01%, and as I said, the dead ones will appear red, while the living ones will remain unchanged. The following is a 3D image of wherein you can distinguish between the dead protoplast and living protoplast. The dead protoplast, they'll appear red. Now the staining method which is used, the third one is chalcofluor staining. The chalcofluor, it helps in determining viability of protoplast. You can know whether the protoplast obtained is living or not by looking for the cell wall formation. So see, the chalcofluor white, it binds to beta-link glucosides in the newly synthesized cell wall, and which can be observed as a ring of fluorescence around plasma membrane. Here the optimum staining it can be achieved when 0.1 ml of protoplast is mixed with 0.5 microliters of 0.1% solution of chalcofluor. The true test of protoplast, the true measure of protoplast is its ability to undergo continuous mitotic divisions and to be able to regenerate into plants. Okay, after the regeneration, after it has been regenerated into plants, it can be cultured on suitable media, on a suitable culture media, and the colonies are obtained. Now you can count the concentration of colonies by using hemocytometer. So this was about purification of protoplast and the staining method. If you found my video to be helpful, kindly like my channel, subscribe to my channel, and please comment down below which which next topic you want me to make videos on. Your one like and subscription means a lot for me. So please like my channel and subscribe to my channel. So until the next video, it's a goodbye. Do take a very good care of yourself. Allah.